This demonstration is going to illustrate a complete synthetic biology workflow using SBOL. In this workflow, we're going to design a genetic toggle switch using parts from the cello dataset that was created by Chris Voigt's lab at MIT. We begin by using the iBiosim software to create two genetic inverters to construct the toggle switch. So for the first inverter, we're going to add a promoter, and we're going to find from the cello parts a promoter that's repressed by the BETL protein. And then we need to add the gene that's going to be coded for. And in this case, let's select this one that codes for the AMTR protein. If you open that up, you can see that it includes a ribosome binding site, the coding sequence for AMTR, and a terminator. So that completes the first inverter. Now we need to move on to create the second inverter. Now the second inverter needs to have a promoter, which is repressed by the AMTR protein. And then we need to have a gene, which creates BETL. That completes the second inverter. Okay, so the next thing we'd like to do is build a model of this. So we select the two inverters to use in our design, select Generate Model, and select the SYMBIOHUB-ProInBiology.org instance where the data is stored. Press OK. And now it's finding all the interaction information, namely what proteins repress which promoters and which proteins are coded for. Select OK. It's returned to us an SBML model that describes the behavior of our genetic circuit. So we can see that here's a model for inverter 2, which is an inverter that's repressed by AMTR, encodes for BETL. Here's a model for the second inverter, which is repressed by BETL and codes for AMTR. And then this one is a model of the composite circuit for the entire design that constructs the toggle switch. We're now going to insert this design into a larger design for testing and simulation. So we add this inverter into our testing structure, like so. Save that. And now we're going to simulate this. We're going to simulate it using stochastic simulation. We're going to perform 20 iterations of stochastic simulation and compute the average result that we get. And this is all done. We have a simulation like so, and you can see that it's acting like a toggle switch, namely it turns on when IPTG is applied, stays on when it's removed, and is reset when ATC is added to it. So this is a nice plot, so I think I'm going to save it. And save that plot for later. Out that goes. Now the next step is I'm going to save this design that I've just constructed into the SymBioHub data repository. So select Export SymBioHub and select the data repository that I want to export this to. Enter my password, provide some information about my design. Like so, and select Upload. And it's now going to upload all the design information I've just constructed, like so. And we go over now to symbiohub.org, and when I log in, and select Browse Design Collections, you can see that on symbiohub.org we have a collection for the AC Synthetic Biology Journal, a Bucellus collection, the entire IGM parts registry, the distribution for 2017, a couple ICE instances, and a link to a private repository within Benchling. We now select Submissions. We can see the genetic toggle switch that we have uploaded. It's in a private submission that only we can see. And here's all the various information we have about it. At this point, maybe I'd like to take that nice plot and add it to my design. So I can attach this plot. I can then describe this design, and I can add my plot to the design, like so, and save that into my little wiki, and you can see that I have that it's been added. So at this point, perhaps I'd like to do some sequence level editing. So let's select the first inverter. That brings up a page describing the inverter. You can see here's a diagram of that. Remember, it's composed of a promoter and this gene. Here's the substructure for that, and if you click on that, it's going to take me over to the Boston University instance of SymBio Hub for the Living Computing Project, where the Cello dataset is stored, and you get a page that describes it, including the reference for the part, as well as some data that's attached to this part. Now we come back here, and we'd like to send this design to the Benchling software for further editing. So it's now copying this design into Benchling. And if we click on this, we can see what it looks like. Inside Benchling, you can see it's been flattened because Benchling only works on flat designs. And if I click Edit, it's going to open up a Benchling window, which allows me to do sequence level editing within the Benchling software. I can click on one of these annotations, for example, and I could change its name if I wanted, like so. If you come over here now and refresh the page, you'll see that the changes within Benchling immediately are reflected within SymBio Hub. Okay, now I'm done with my sequence level editing, so I'm going to copy this from Benchling back to SymBio Hub, and I'm going to copy it into my collection, like so. And so now this is showed up within my collection here, and there's the sequence level editing design. Now at this point, perhaps I've actually constructed this in the lab, and I want to keep track of the plasmid I've constructed, and I can use the ICE registry for that. Select Send to ICE, select an ICE instance, send it there. Now it's copying it into the ICE registry. And so here is the genetic inverter within ICE, on the ACS Synthetic Biology ICE. So here it is there, and if you come select there, you can open it up within the ICE instance, and you can view it in ICE. And if I'm happy with that, I can copy it from ICE back into SymBio Hub into my existing collection, like so. And I'm back to my original collection. Okay, so at this point I think it's pretty good, and I'm ready to publish it. So I want to submit my paper, and I want to share this privately to the reviewer. So I can click Share, and this creates a shareable link. So if this link here is not accessible by anybody but me, but if I use the shareable link, just like in a Google Doc, and select that link, this can be opened by anybody. So if we pass that link to the publisher, then the reviewers can open up your design and see the design information about your design. Once your paper's been published, perhaps you'd like to add the reference into here, a PubMed ID, like so, and that will look up the reference for your paper. And if you select Make Public, you can then decide where you want to publish it. I want to publish it into the Synthetic Biology collection. Select Make Public, and it copies it over into that collection. And now I can open this here, and I can see the genetic toggle collection for my design, and all the design information has been published, and anybody can see it at this point and search upon it and use it in further designs of their own. And that concludes our demonstration.